Thanks to my co-speakers, Iyad Mashal, Dr. Muna Al-Hadidi, and Dr. Shmuel Suman for the fascinating talks. Certainly, as some of the most water-scarce countries in the world, we share interest in utilization of non-drinking quality water sources for irrigation, and we can see the need for knowledge and tools for making sustainable decisions regarding such use. This is certainly a topic that could drive us forward together. Our third and final session regards soil and plant nutrition management. We will first hear from Dr. Hakim Boungao of the African Plant Nutrition Institute in Morocco, who will talk about nutrient management of olive orchards. Next, Professor Lina Yusuf of Khalifa University in Abu Dhabi, the UAE, will share work on biochar as a soil amendment. Finally, Dr. Ron Arel of the Gilad Research Center of Israel's ARO Volcani Institute will discuss allocation of calcium to fruit and connections to abiotic stress causing factors and the combined effects on crop physiology. Let's go to the session.
Okay. First of all, I would like to thank the Minister of Agriculture and Rural Development and Volcanic Institute of Israel for the invitation for this important scientific conference about sustainable agriculture in area. My talk today is about the olive sector in Morocco and mainly about the challenges of nutrient management of olive orchard. Olive orchard, as you know, is an important crop tree in our country, represent almost 65% of the total tree crop area in Morocco, and 5% contribute to the national agriculture gross domestic product. Most of the olive tree in Morocco are concentrated in the center of the country and also in the north. Even in you can find, except the Sahara, in all the country olive tree. And the olive sector in Morocco is divided mostly in olive oil and the table olive. Olive oil is the most dominant with 75%, and the rest is represented by the table olive. Also, the olive sector in Morocco contributes significantly to the employment. And in the last statistic showed that olive sector contributes to almost 380,000 permanent jobs, with almost 20% of workers are women. The olive farming type in Morocco is divided into three dominant olive farming type in the country. The most dominant is the traditional plantation with low densities, often with ancient trees and typically with few or no chemical input. The other one is the semi-intensive traditional plantation, mostly under irrigation with systematic use of fertilizer, but without considering the needs of the tree. And the last one is the intensive, modern, super high density plantation introduced in the early of 2000 and managed under irrigation with high input application. Also, one of the characteristics of the olive sector in Morocco is the dominance of the intercropping, mostly cereals like wheat and barley, but also maize, other autumn sowing crops such faba bean, lentil, pea, and chickpea. And also we have in irrigated area, onion, cucurbice crop, but also the forage crop, alfalfa. In the last decade, Moroccan government gives more attention to the olive sector in the country. And the Green Morocco plan, one of the big agricultural plan in the country between 2008 and 2020, contributes significantly to the olive sector. And as you can see in the two figures, the total area of olive orchard increased from less than 600,000 hectares in early 2000 to more than 1 million in 2020. In the same evolution, the production of olive increased significantly with less than 500,000 tons in early 2000 until more than 2 million tons in 2020. But the increase of the production of the olive in Morocco is not due to the high yield, but just in the increase of the olive orchard area, because the olive yield in Morocco still low with almost 1 ton per hectare under rainfed condition and 2 ton per hectare under irrigated condition. Nutrient deficiency and imbalanced fertilization remains one of the main contributors to high olive production, because we need adequate nutrition for the growth, for the flowering, but also for the better fruit development, and in conclusion for the production. The most research over the world or over Mediterranean areas showed that olive trees respond well to fertilization. With the exception of water, nutrients are the environmental factor that most contribute to tree growth. Although the olive tree is considered a rustic species with a great capacity to survive and produce in low fertility soils, it must have an adequate level of nutrition to perform the essential functions of vegetative growth and reproductive development. Now, the aim of fertilization is to maximize nutrient uptake efficiency and to increase its content in the tree. Nitrogen, phosphorus, and potassium are the most frequently macronutrients that limit the plant growth for the case of olive tree. It has been demonstrated overall that the lack of nitrogen is the most common nutrient deficiency in olive tree growth, followed by potassium, because you know that potassium is required at later stages of crop growth for repairing the fruit, but also less than important than nitrogen, phosphorus. Also, phosphorus fertilizer has become more common with the increase in intensive olive growth in this system. In terms of micronutrients, boron have shown a key role also in olive productivity. In some research in Morocco have demonstrated this finding, because boron plays an important role in olive fruit set, oil content, and also oil quality. Some research undertaken in Morocco, in some region of Morocco, have demonstrated that 100 of olive orchards were deficient in nitrogen and more than 80% deficient in potassium and less than 40% in phosphorus, which demonstrated the role of nitrogen, potassium, and phosphorus for olive tree in Morocco country, but also in Mediterranean area. Now, the use of fertilizer in olive orchard depend on factors for such rainfall, irrigation, farmers' income, and local tradition. Because you know, olive orchard in Morocco is found in both system, in rainfed, in irrigated system. Until now, in olive orchard in Moroccan system, we don't have a standard practice for fertilization in olive farming, because we have a high variability of olive farming type between the region of Morocco. And farmer fertilizer practices in Morocco are in general traditional, based mostly on organic fertilizer with few complements of mineral fertilization, mostly nitrogen, without any specific knowledge in the best technical practice. More attention is given to mineral nutrition in high to super high density olive orchard under irrigated condition, with the introduction of fertilization based on nitrogen, phosphorus, and potassium as major sources of nutrients. Now, maintaining and improving nutrient status of olive trees is a key for the sustainability of olive in the country in Morocco. The first research on nutrient management in olive orchards in Morocco started with the International Plant Nutrition Institute in 2013, followed by the African Plant Nutrition Institute in 2019. The first research were based on the diagnostic of nutrient status of olive orchards based on soil analysis, foliar diagnostic, and visual examination of the symptom of the deficiency of nutrients. The first finding showed that soil analysis is not accurate enough to be used to diagnose fertility in olive orchards. Also, we have assessed method of analysis such as removal and leaf analysis for nitrogen, phosphorus, and potassium. The both method, removal method and leaf analysis were compared to the farmer practice, fertilization farmer practice and the control without any application of fertilizer, because in some traditional also olive orchard in Morocco, most farmers, they don't use fertilizer. This finding, even in off-year or on-year or also in irrigated area or rainfed area, showed a significant effect of the both method, leaf analysis method and the removal method, compared to farmer fertilizer practice or the control. Both methods, they have increased significantly the olive yield in the both system, irrigated or rainfed or in on-year or off-year. For example, over 4 years, we have found that in rainfed area, olive fruit yield were improved respectively by 60% and 44% by applying leaf analysis method and crop removal method. And also in irrigated area, the fruit yield were improved significantly compared to fertilizer farmer practice or the control. The same finding and the same effect were found in the effect in oil content. The leaf analysis and removal method were significantly higher in terms of oil content compared to farmer fertilizer practice. Comparing the two methods, the leaf analysis or the removal method, even for the case of the effects in oil content or the olive yield, we showed that leaf analysis method give more significant effects compared to the removal method. Also, we have assessed the two methods compared to farmer fertilizer practice and the control in terms of oil quality, and both methods we have found that they increase significantly the total phenols for oil, even for the case of on-year or on-year. But this method cannot be applied within the adoption of the 4R nutrient stewardship, which means the application of the right source of fertilizer at the right time, at the right place. This concept of 4R nutrient stewardship was adopted with the IPNI, International Plant Nutrition Institute, for several other crops. Now, the new challenges of nutrient management of olive orchard in Morocco is to increase the production in terms of quantity and quality and adaptation to climate change, and also for the protection of natural resources and the conservation of landscape. Closing yield gaps is the main challenge for olive production now in Morocco. As you know, the population, not just in Morocco, but over the other country is increasing, but the surface area is limited. Why we need now to increase the yield per hectare. For that, for the case, for example, for the olive orchard in Morocco, we need to characterize the olive potential yield per region, because some regions have much greater potential than others to support high yield in a sustainable manner. Also, we need to understand the response of olive varieties in different ecological condition to enhance the development of better nutrient management program. Also, facing the climate change presents an important challenge for sustainable nutrient management in olive orchard, because the most vulnerable category for climate change, as you know, are smallholder farmers. And this category of farmers, they grow mostly olive orchard. We need also to know that increased frequency of warming and drought events are some of the constraints that growers will have to deal within the upcoming decade. Also, in rainfed orchard, water is very often the limiting factor for nutrient availability and uptake. Why understanding the main response of mechanisms adopted by the olive tree to cope with drought is crucial to improving nutrient management strategy. Also, we need to have best practice for minimizing negative impact also in the environmental practice, such cover crop with minimum tillage or no tillage system to avoid nutrient losses in intensive olive orchard is highly recommended. Also, we need to recommend the introduction of some leguminous as cover crop to improve the profitability and the sustainability of rain-fed olive orchard. Some also controlled application of olive meal waste can provide significant amounts of potassium and phosphorus to the soil and partially to replace fertilization with these nutrients. Experience in Morocco to assess the effect of the introduction of legumes as intercropping showed that with the association of olive orchard with some legumes, such faba bean, for example, increase significantly the olive yield compared to olive associated with cereals crops such wheat or olive in monoculture. Also, we need some measures to increase soil organic content to improve soil fertility in growth. Pruning residue should be incorporated in soil rather than burning. Also, this practice is becoming more common in some Mediterranean region, but this practice is not common, for example, in the case of Morocco. Also, one of the recommendations is mulching, also pruning residue instead of burning for improving soil fertility. So it is advised that the variability in soil fertility in relation to the distance of olive tree should be taken into account for increasing the efficiency of best soil management practices in the irrigation, because the research demonstrates that soil chemical and microbial property were significantly higher on the tree crop compared to outside the canopy, because we are now applying what I said intercropping in olive orchard system in Morocco. We need to take into account this variability between the soil fertility under tree crops and outside the crop. Also, empowering research domain and nutrient management for olive orchard is very important in Morocco. Research and plant nutrition by national institution on olive tree are not considering nutrient research into their priority, because most of research programs are mostly focused on genetic and pest management. We have also an absence of fertilization standard for olive tree adapted to each agroclimatic region in the country. We need also support fertilizer related research, education and extension projects. These efforts include improving the efficiency of nutrients, promoting more effective use of fertilizer and developing precision technology. Also, it is recommended to promote the use of 4R nutrient stewardship. Existing and future supported research for improving nutrient use efficiency, coupled with the application of the 4R, will help solve the nutrient challenge. And also, we need to improve the understanding of best nutrient management of olive orchard. In Morocco, the extension of new plantation outside their habitual ecosystem need for the research for understanding the role of the new environment in the response of olive orchard to the fertilization. Also, the adoption of improved best nutrient management practices based on agroecology can help build resilience to climate risks in olive. And by the end, we need a strategic collaborative program for Mediterranean countries to communicate and exchange the main findings and experiences, not only in the complexity of the nutrient challenges in olive orchards, but in the global value chain. Thank you very much.
Hi, everyone. My name is Lina Youssef. I'm an assistant professor of chemistry at Khalifa University. I'm also co-founder of Delarta, the Outdoor Living Laboratory. This is a startup that focuses on maximizing the use of plant and soil resources in arid regions like the United Arab Emirates. Today's talk is about biochar. So a little bit of background on myself. I'm a multidisciplinary scientist. I've dabbled in quite a different range of projects, but I spent a substantial amount of time working on biochar and characterizing biochar for its use in agriculture. So just as background on Abu Dhabi is one of the 7 emirates in the United Arab Emirates. It is the largest emirate based on area size. So it's about 87% of the total land area of the country. And Abu Dhabi actually has a soil map. They have 2 scales, an extensive and an intensive survey. So higher resolution for the intensive survey. And you can find additional detail and also play around with data through this website here. But just a general overview, soils in the UAE and in Abu Dhabi in particular are fall into 2 of these soil orders, the intersoils or iridosols. Intersoils lack any soil profile developments, as you can see here, and they're predominantly found inland. And then you have the iridosols, which are mostly found in coastal areas. And if you want to break down the classification of soils further, there's about 64 series that have been identified. So when we're thinking about agriculture with respect to outdoor field-based agriculture, soils are typically low in quality. And the reason for that is the genetic soil forming factors are not favorable. Due to the warm climate, the country's in a hyper-arid region, which results in low vegetation cover. The texture of the soil is sandy, so it has low water retention. There's also high amounts of salt precipitates. So soils are typically either saline or sometimes saline sodic. And also the pH of the soils is quite alkaline, which limits their fertility. And there's very low organic carbon in these soils, so less than 1%. So again, due to all of these factors, soils are naturally not suitable for agriculture without extensive irrigation and input of fertilizers, et cetera. So to improve soil quality, there's several approaches. You can improve soil texture to improve water retention. I mean, this is quite important in arid regions. You could also improve soil fertility by improving pH, and that's through pH conditioners to achieve a more nutrient balance. And then also we can work on reducing the salt content. So there's different mechanisms to do that, leaching out if in the case of sodic soils, or phytoremediation is another approach. Another thing we can do is increase the organic carbon content of the soil, and that's through the application of organic amendments. The problem is a lot of these organic amendments have a very short residence time in the soil, so it's quite costly for these farmers. They have to apply these organic amendments on a regular basis. And another question is, are all organic amendments equal in terms of their benefit? So this is where biochar comes into the picture, and it gained a lot of popularity, and there's a lot of hype around biochar, specifically in the past 2 decades, because biochar is believed to have high recalcitrance, as in it has longer, its longevity in the soil is quite good, well, at least believed to be so. And there's a lot of hype around it in terms of improving water retention in soil, et cetera. So the question is biochar always effective? And this is just a little bit of general background about what biochar is for people who are not in the biochar research area. Essentially, biochar is just a fancy word for charcoal that's made from plant material. And the application of biochar to soil is not a new practice, although it kind of surfaced in the past 2 decades. but it's actually over a 2,000-year-old practice. And if we go to the Amazon basin, we see terra preta soils. So terra preta is just a Portuguese word for black soil. And these are a result of natural, they're natural in presence, and they come from forest fires. So natural forest fires, and then of course, all that carbon's buried in the soil. If we look at the number of publications just in the last 10 years, there's over 30,000 publications available. A lot of this research is geared on looking at what potential biochar has in terms of increasing water quantity and quality, improving nutrient cycling and bioavailability. How does it support soil biodiversity? Does it improve crop yields in agriculture? And then also from a greenhouse gas emissions perspective, can we use biochar as a tool to create a carbon negative system where most of the carbon stored in plant biomass, instead of it being decomposed and released back into the atmosphere, can be stored as part of the soil carbon pool for a longer time period. So again, this kind of visual to show you what biochar can do. So this is 0.5% organic carbon in the soil. This is about 9% with the application of biochar. And you can see here, the result is you have an extensive increase in not only density, plant density, so this is corn, but also the plant height is also much higher, larger, as you can see here. And if you do a quick search on the biochar market size, you'll see that it's worth a little over $3 billion. And it's growing at an annual growth rate of 13.2%. So it's quite a hot market. And of course, all of these companies are capitalizing on different types of feedstock. So you could see this is just a snapshot of some of the products that are available in the market. They're sold in different, based on volume or mass. And of course, there's different price points depending on what kind of biochar it is and what kind of marketing each company is doing. So for example, for carbon gold, you could see that they're being a little more creative. They're actually doping the biochar with microbes and trace minerals. So there's an added benefit there for its use. So biochar is a function of, to come up with a biochar from plant biomass, this is a function of temperature, heating rate, time, and feedstock. So you can be super fancy. And by the way, biochar initially was a byproduct of pyrolysis. So this here is a pyrolysis reactor in which plant biomass is converted predominantly to biogas or bio-oil for energy. But then you have some charcoal that's produced or biochar produced. And this is where all the hype started, where they're looking for an output for this byproduct. And you can play around with these functions here. So the major one being pressure and heating rate. This really kind of controls how much of the biomass is converted to gas, oil, or charcoal. Then you could go to a low technology like an open field or open air kiln. As you can see here, this is a low technology where you're essentially just burning the biomass. And the purpose of this is to convert all of it into biochar, as you can see here. So there's a lot of companies that are capitalizing also in these technologies on pyrolysis reactors, as well as different formats of these kilns. A little bit about the research that I did with my group. So one paper that I looked at is characterizing biochar quality. So here I looked at two different feedstocks. One is date palm fronds, the other one is roadgrass. And the reason we chose these two types of feedstock is date palm fronds is a problematic waste in the United Arab Emirates. So we wanted to see if we can create an avenue for its use. And then rosegrass is a type of grass that's used as animal feed. So we wanted to see if it could be converting some of the road grass that's cultivated on these farms as part of a management practice, where some of it is converted to biochar and applied to soil to improve soil quality for further use. So the point of this is you have two feedstocks converted to biochar under similar conditions. So 400 degrees Celsius, heating rate of 5 degrees Celsius per minute for a duration of 11 hours. And what happens is you could see there's a real difference in terms of the macrostructures of the original biomass converted to biochar. So what we looked at is we looked at measuring the diameter of these pores. So first of all, in both types of biomass, it's predominated by macropores. So these are pores that are greater than 0.5 microns in diameter. So what we did here is we essentially measured the pore diameter and plotted it based on range. And we wanted to look at the changes in the curve. So you can see here with date palm fronds, it seems to be quite sensitive to heat. So once it's converted to biochar, you'll notice there's a lot of gunk here. So these are volatiles that are trapped in these macropores. But also if you look at the pore size distribution before and after, you see that there's a major shift in pore size distribution where the pores are getting larger and there's more of a bell curve occurring. In contrast with the Rhodes grass, you see that there's very little change in the porosity distribution before and after. So this tells us that it's more resistant to thermal decomposition. If we're going to look at characterizing the biochar itself, we can see that Rhodes grass has a higher surface area, and this is corroborated by the smaller pore-sized macropores present in the material. And again, remember the gunk here that's seen, you could see that there's a high % of volatile carbon in the date palm. So this is what this is in contrast to the Rhodes grass. Another thing to assess the stability of biochar with respect to its residence time, we look at the carbonization ratios of H to C and O to C, so oxygen and hydrogen to carbon. So the smaller this ratio is, the more recalcitrant the material is. So we can see here with respect to the oxygen to carbon ratio, the date palm is much higher than the Rhodes grass, which tells us that it's less recalcitrant. Another study that I did with a group in Australia is we looked at anaerobically food waste digestate. So this is the raw material. And then we converted it to biochar at different temperatures. But the point of this is we wanted to know, does the biochar perform better than the raw material with respect to agricultural use? So we used the soil from the UAE, a typical soil here. So the baseline conditions, the pH 7.9, with an electrical conductivity of 752 microsiemens per centimeter. And one thing we noticed is the application of biochar. This is the 500 degrees Celsius biochar. There was a reduction in plant available water in contrast to the raw material. But when we looked at the germination index of biochar or raw material extract, we noticed that the biochar extract had a beneficial effect on germination in contrast to the raw material. Another thing that we noticed is the electrical conductivity of the soil actually declined with the application of biochar, whereas it increased with the application of raw material in a consistently linear manner. And again, the pH of soil treated with biochar was quite high in contrast to the pH of the raw material. So again, there's a mixed benefit. You could see there's an increase in pH, but there's a decrease in soil salinity. It's good for germination index, but it's really poor for plant available water. So this is a really nice paper that looked at 16 studies. So they did a meta-analysis on 16 studies and 77 treatments, different types of feedstock, application rates, and plant yields, either plant biomass or grain yield. And one thing that they realized is the most benefits are observed on acidic and neutral pH soils, less so on alkaline soils. And the greatest positive effect was observed when you use an application rate of 100 tons per hectare. And typically there was a 10% increase in plant yields. So I'm going to show you two studies. The first one here is looking at two different types of biochar, one that's generated from cocoa shell, one from rice husk. And this was applied at different rates over 5 different seasons. This research group measured maize, so corn. They measured grain yield in tons per hectare. And what they noticed is in this acidic soil, it's quite toxic. There's aluminum toxicity, which prevents plants from growing. So when they applied biochar at a rate as low as 5 tons per hectare, they noticed there was an increased benefit only when they used cocoa shell biochar. Whereas they didn't see the same observation in the rice husk biochar. So another observation they made is that the application has to be applied every 5th year. So you can see here clearly with the 15 tons per hectare that after the 4th year, the yields decline. So it's advisable to reapply biochar during that time. Another study done in Germany also looked at biochar, but this biochar was made from a different feedstock, hardwood. And the point of this study is they didn't observe any positive effects in terms of crop yield. So looking at plant dry mass, so this again is corn, they noticed actually there was a decline in yields of plants with the application of biochar. As you can see here, in plant height, the controls performed much better than the application of biochar. So the take-home message here is biochar could benefit soil and the environment, but you really have to really study both the soil and the biochar before making a decision on this. So the answer is biochar friend or foe. It really depends, and it requires a careful analysis prior to use. And that is the end of my talk. Thank you so much.
Hello, everybody. My name is Ran Rel. I'm a researcher at the Gilat Research Center. I'm studying plant nutrition, and I'm specifically interested in plant responses to abiotic stress and whether we can enhance the resilience of plants by optimization of mineral nutrition. Calcium is very important in terms of plant response to abiotic stresses, and especially in fleshy fruits, where calcium is the main skeleton of the plant, of the tissue. It's the cement that binding the cellulose microfibers and create the wall cell rigidity that is necessary for firm fruits. Calcium is also very unique in its mobility in plants. It's only mobile in the cell line, unlike most other nutrients, and therefore it tends to accumulate in the transpiring organs. In the slide in front of you, you can see how healthy plants looks like versus nitrogen or magnesium deficiency, where you see the deficiency symptoms at mature leaves, while the young developing tissue is healthy. In the calcium deficiency, it's the opposite. The mature leaves look nice, while the young tissue is very restricted by the calcium deficiency. And the reason is that calcium is not mobile in the plant, and therefore once we stop to supply calcium, the growing tissue is severely damaged. So this is very unique. And when it comes to fruit, this is specifically important because fruit does not transpire at all. And therefore, a typical plant, in this case, tomato, you can see that fruit calcium concentration is about 20 times lower than the leaf concentration. And that causes many disorders like the blossom end rot that is very common in tomatoes and pepper, where you see the tissue collapse because there's not enough calcium to build the cell wall. So this is very common in tomato, of course, and in pepper, but the same physiological disorder, different symptoms occur in apple, that's called the bitter pit, or in lettuce, curly leaves. These are all symptoms of calcium deficiency in organs that do not transpire. I was surprised to learn that tubers of potato are pretty much the same. When it's hot or dry, the tubers have very low supply of calcium, and what happens is these browny spots that cause internal rust sometimes, and that occurs in the heart of the tuber and a result of the low calcium supply to the tuber. In Israel, for example, outside the winter, some varieties cannot be grown because of internal rust. In a classical study, Jitan Palta and colleagues showed using labeled calcium that when the plant is supplied with calcium to the main root system, all of the calcium goes up to the shoot. And the influx into the tuber occurs only from the small roots that found on the stolons. So supply of calcium is very, very limited. Now, how is that related to environmental stresses? We came with a hypothesis that during the day, due to stressful conditions in desert environment, the rhizosphere will be drier. The roots absorb water, and therefore salinity around the root system increases just in the vicinity of the roots, and that causes very hostile conditions for nutrient uptake. While in the night, the opposite occurs. There is diffusion of water from the bulk soil to the roots, and there's diffusion of salts from the roots back to the bulk soil, and that creates better conditions for nutrient uptake. And we wanted to study whether plants acquire nutrients more in day versus night, and specifically calcium, which is important. But soil has a lot of calcium, so how can we know when the calcium is being uploaded into the fruit? So we use strontium as a tracer. Strontium is very similar to calcium, just next to it in the periodic table. They share many chemical properties, and it was shown to be a good tracer. We tested it in a long-term experiment with tomato, where we supplied either calcium, strontium, or no calcium and no strontium, and we found that the biomass was comparable, and that strontium prevented the blossom end rot at the same level as calcium. That means that it's being uploaded to the fruit and accumulates in the fruit in the same level and prevented blossom end rot. And the nutrient concentration of strontium and calcium that you see here is comparable. And therefore, we concluded that it is a good tracer for uptake and partitioning in the plant. Later, we tested the uptake in day versus night in hydroponic system that you can see here at 4 different concentrations of strontium. And you see that to the shoot, there is minor differences between day in the yellow and night in black. So it's comparable, a bit more uptake to the shoot in the day compared to the night. While we look at the uptake rate in the fruit, we see a tremendous difference. And we see that in the night, there's roughly 4 times more strontium uptake to the fruit compared to the day, where it's very, very low amount of strontium was transported to the fruit. Now that was in greenhouse. We wanted to further investigate the effect of severe abiotic stress. So we put the plants, new set of plants in greenhouse. We increased temperature, decreased humidity, and accelerate transpiration by fans, and tested how much strontium was uptake to the shoot or to the fruit during the night and the day. At this case, we see that in the day, the strontium uptake was double than the night in the shoot, but in the fruit, well, you see here that the only time that strontium was found in the fruit was in the night, and in the day, the uptake was nearly 0. So under conditions of severe stress, the night is the only time that strontium is being uptake, although that in the day, the total uptake is higher. To summarize this set of experiments, we did different manipulation, and here we plot the portion of calcium or strontium as analog uptake in the fruit divided to the whole uptake versus transpiration rate. So during the day or when it's hot, transpiration rate is high, and hardly any calcium is being transported to the fruit. During the night or in the winter or when humidity is high, transpiration rate is low, and then the portion of calcium that's being transported to the fruit is much, much higher. And maybe we can use this data to develop better strategy to supply calcium to fleshy fruits in stressful conditions. A second example is with potato. We grow a lot of potato around here in the Negev area. And recently, farmers complaints or the consumer complaints on this disorder, peel disorders. They just don't look so good. It's not a disease, it's not an insect, it's just a visual defect. And we immediately suspected that it's related to calcium because there is data that's showing that the more calcium we found in the tuber, the less it's susceptible to all kinds of defects. The second reason that we think calcium and magnesium are highly relevant, because in the past 10 years, we have increasing utilization of desalinated water. This desalinated water goes from the domestic use to the farmer, and nowadays farmers in the Negev uses water that used to be from the desalination plant and have very low calcium and magnesium concentration, significantly lower than what they used to have. And I collected hundreds of soil samples from the farmers in the Negev and plotted the date from the start of using desalinated water to now versus the calcium and magnesium in the soil. And we see a significant decrease in the extractable calcium and magnesium in the soil in the Negev, meaning that the change in water quality is very likely change the composition of nutrients in the soil. We tested at 10 different sites with and without supplying calcium and magnesium. And what we found that not the fertilization, but soil type had the most important effect on the composition. In sandy soil, as presented here, the concentration of nutrients in the potato was significantly lower, roughly half than the loamy soil, the more clay soil that has accumulated much more calcium and magnesium. When we look at these visual score of the potatoes, we see that the different soils had the same score immediately after we picked the tubers, but after 1 month in storage, in the sandy soil, there's a very significant decrease in the score from 3 to 2. 2 is very low quality, while in the loamy soil, where they had more calcium and magnesium, there's hardly any decrease. This is what you see here, the change from harvesting to after storage. So eventually, we concluded that in sandy soil, the low supply of calcium and magnesium might be contributing to the low quality of the potato peel, and that's very probably a result of the change of the water quality. To summarize, we saw that that calcium is very important in fleshy fruits, and we can use strontium to monitor the transport of calcium in the plant. We see that abiotic stress enhances the calcium uptake to the shoot and diminish the calcium uptake to the fruit, and therefore supplying calcium during the night, that's the most important issue, and that should be tested in the field. We see that in the potatoes, low supply of calcium and magnesium in sandy soil might have an effect on the peel quality of a potato, and therefore in these types of soil, we need to consider adding calcium and magnesium to the irrigation. I would like to acknowledge the funders is the Israeli Vegetable Board that supported the study on tomatoes, and the CFPN, the ICL, and Arrow initiative that supported the potato research. The students that carry out most of the work, Peter and Noam, and my lab members. Thank you.
Big thanks to Dr. Hakim Bungao, Professor Lina Yusuf, and Dr. Ron Arel for the great, fascinating presentations. We're now going to move to a panel discussion where all of today's speakers are invited to answer your questions and for us together to address issues and ideas for promoting regional collaboration. What I'd like to do is, one, please continue to write your questions or comments in the chat. Two, those of you who are panelists in the panel room, please turn on your cameras. I'm gonna assume, unless you tell us otherwise, that if you're not seen, you're not interested in being in the panel, and we will move you to the general population.